Hello my dear students, this is Dr. Pooja, your biochemistry mentor. Now in this video we are going to discuss the qualitative tests for carbohydrates. So let's start with the discussion. Qualitative tests for carbohydrates. Now we all know that how do we define the carbohydrates? They are simply the polyhydroxy derivatives of aldehydes and ketones. Right? And on hydrolysis they are going to give aldehydes and ketones. Now this we have already discussed in the chemistry of carbohydrates. Now today's topic is qualitative analysis. Qualitative means that just by seeing the color of the solution, we can say that carbohydrate is present in this. This is a very important topic, and basically first of all, if a sample is given to you, you have to first of all find out whether it consists of a carbohydrate or not. It can be a protein, it can be a fat. So first of all, we have to go for a confirmatory test for a carbohydrate, and once it is confirmed, then we can proceed for the further practicals, right? So, and we have studied about the classification of carbs, that we can divide carbs into basically three categories or three classes as per the practical application. One is the monosaccharides. Second is the disaccharides, and third is the polysaccharides. As per practical application I am saying, otherwise so we can classify carbohydrate into four major classes. Oligosaccharides also come. Okay. So first of all, we have to see whether the sample is a carbohydrate. So for that we perform Molisch tests. So if Molisch test is positive, I'll just show you the schematic plan first of all, then we will go with the explanation of each and every reaction. If the Molisch test is positive, that means carbohydrate confirmed. Now once a carbohydrate is confirmed, then we have to see whether it is a monosaccharide or a disaccharide or a polysaccharide. For that we perform iodine test. Right. Up simple, what we do is we take the sample, we put iodine solution into it. So what will be the result if I say that the test solution gives a yellow color or the native color of iodine? If it gives native color of iodine, that means it can be a monosaccharide or a disaccharide. But if it gives bluish violetish color, blue or violet color, that means polysaccharide or starch is there. Polysaccharide or starch is present. Okay. So if polysaccharide is there, we are going to perform a confirmatory test for starch. But if it is giving the native color of iodine, we are going to see whether it is a monosaccharide or a disaccharide. Now to differentiate between a monosaccharide and a disaccharide, we go for Barfoed's reaction or Barfoed's test. Barfoed's test. Now again, while performing the reaction with Barfoed's reagent, if the precipitation appears immediately, right? If the precipitates, or you can say red PPT, comes immediately, it's monosaccharide, right? And if the precipitates come after heating, precipitation after heating or after some time, then it is disaccharide. Because obviously when we are going to heat the solution, so the bonds will be broken or disaccharides will be converted to monosaccharides, which will react with the Barfoed's reagent. Now we know whether it is a monosaccharide, whether it is a disaccharide. Then the most important test for this is, now we have to find out whether the sugar is reducing or not, and that's a very, very important test, and it is actually a test of clinical importance also whenever we have to check the presence of reducing sugars in urine, because this is one of the very important abnormal constituent in urine. Okay, so if we talk about the test which distinguishes between reducing and non-reducing, that test is known as Benedict's test. Or I would say this test distinguishes between a reducing and a non-reducing sugar. So if Benedict's test is positive, there can be only two reactions, positive or negative. So if the reaction is positive, we say that the reducing sugar is present. Reducing sugar. And if the test is negative, it is a non-reducing sugar. So Benedict's test distinguishes between reducing and non-reducing sugar, right? Now what we have, and there are certain specific tests also. This is what, suppose if you've been given a sample of glucose and fructose, because we know that glucose is aldohexose and fructose is a ketohexose. Both are going to give the same reactions till Benedict's reagent. So there has to be some test which can distinguish between an aldose sugar and a ketose sugar, and that test is Seliwanoff's test. Seliwanoff's test, right? Or Seliwanoff's test. Now this test will be given only by ketohexosis. So if this test is positive, again there can be two, positive or negative. So if the test is positive, it is a ketose sugar, or I can say ketohexose. And if this test is negative, it is a aldose sugar. So this is the schematic plan which we are going to follow when unknown sample is given to us. Okay, so let's quickly go through this. When an unknown sample is given to us, we have to first of all check whether it is a carbohydrate or a protein or a fat. So if it is giving a positive Molisch test, that means the presence of carbohydrate is confirmed. Then we have to identify whether it is a monosaccharide, disaccharide or a polysaccharide. For that we perform iodine test. So iodine test, if it is positive, that means the sample is a polysaccharide. But if it is giving the native color of iodine, it's not giving blue or violetish color, that means it can be a monosaccharide or it can be a disaccharide. Then we have to perform Barfoed's test. Barfoed's test distinguishes between a monosaccharide and a disaccharide. And if it gives red precipitates immediately, that means it is a monosaccharide, and if it gives after heating, then it is a disaccharide. Then comes the most important test, which is Benedict's test, which distinguishes between a reducing sugar and a non-reducing sugar. So if this test is positive, that means the sample is a reducing sugar. But if this test is negative, the sample is a non-reducing sugar. And then we have to identify or differentiate whether it is a aldose or whether it is a ketose. For that, Seliwanoff's test is there. Okay. Then after this, then we come over to what is the principle or what is the rationale behind this Molisch test. Now, this Molisch test is actually the alcoholic solution of alpha-naphthol. Simple. Alcoholic solution of alpha naphthol. And if we talk about the principle is it leads to the formation of furfurals. So there is furfural formation. Right. And since it is an alcoholic solution of alpha naphthol, then we add concentrated H2SO4 along the sides of the test tube. I'll just show you the picture also. The reaction will be this. All sugars in the presence of the oxidizing agents have a tendency to form the furfurals, because obviously they will undergo Molisch or you can simply say that they will undergo oxidation, right? So if we talk about glucose, you can see that it is going to form this hydroxymethyl furfural. Okay, so basically furfurals are the products which are synthesized whenever a carbohydrate undergoes oxidation. So be it glucose, be it fructose, be it any carbohydrate for that matter, it is going to give a positive Molisch test. So it is going to form hydroxymethyl furfural and which will condense. You can see hydroxymethyl furfural which is condensing with alpha naphthol, leading to the formation of this type of complex, because it is leading to basically dehydration. And then we add concentrated H2SO4 along the sides of the test tube. And the moment we add concentrated H2SO4 along the sides, it gives a ring. This type of ring is obtained whenever we perform Molisch test. Very important. Clear? So formation of a ring is indicator of, there is one more ring formation, that is Rothera's test. And Rothera's test is also a very important test because it helps in the, you can say, diagnosis of the ketone bodies which are coming in urine. That ring and this ring is totally different. So don't get confused with that. Molisch test is given by all carbohydrates. It is not that we are detecting some abnormal constituent in urine. For that, Benedict's test is there. Okay. Molisch test is simple. For that matter, all glucose molecules, all carbohydrates will undergo oxidation, forming hydroxymethyl furfurals which will condense to form a colored compound in the form of a ring. Okay. So this is Molisch test, confirmatory for all carbohydrates. Then comes second test is starch or iodine test. Now if we talk about iodine test, iodine again simple concept is there, only given by polysaccharides. Given by polysaccharides and mainly starch, because we do not, physiologically important polysaccharide is glycogen, but we do not give glycogen. So starch will give a positive iodine test. And if we talk about the confirmation of this, what we do is simply we take a solution and we add iodine into it. So if it gives violetish color, right? Suppose if it is giving a violetish color, we can say that bluish or violetish. That means starch is present, as I have written here. So what we do is we have to just heat this solution. So when we heat this solution, color will disappear. Color disappears. Why? Because heating से क्या हो जाएगा? Iodine will come out from the glycosidic linkage. Iodine is released from glycosidic linkage. And on cooling, the color will again appear. On cooling, color appears. So this becomes a confirmatory test for starch, so that we can say the sample is polysaccharide. Now, what if the sample is giving, you can say, a color of iodine? That means it is not starch. That means it can be a monosaccharide or a disaccharide. So for that we perform Barfoed's test. So Barfoed's test distinguishes between monosaccharides and disaccharides. Between mono and disaccharides. And again, it is simple. What we do is we take a solution and we add Barfoed's reagent. But here we can say this Barfoed's reagent is or you can say the reaction is performed in the acidic medium. Reagent in glacial acetic acid. Right. And it is going to give red precipitates of cuprous oxide. So if red precipitates appear immediately, we can say the sample is a monosaccharide. But if precipitation is there after heating, because what will happen when we heat a solution, the bonds will be broken and disaccharides will be broken into monosaccharides. So automatically they are going to give the same color. So this is Barfoed's test. Then we come over to the most important test, Benedict's test. Clinically this test is very, very important. Because Benedict's test, first of all, it is a test to distinguish between or differentiate between reducing and non-reducing sugars. Right. And this is a test which can detect the presence of abnormal constituents, because I have already explained what are reducing sugars. This you should know by heart. Reducing sugars are the sugars which have a free anomeric carbon atom. Meric carbon atom, which is C1 actually. Very, very important. Okay. So what happens is this Benedict's test is also known as a qualitative test, or it is also known as semi-quantitative test too. Qualitative test too it is there, but it is quantitative also. And that too semi-quantitative. What do you mean by semi-quantitative? That just by seeing the color of the solution, we can say that the amount of sugar is present. So obviously if sugar is coming in urine, that it is indicating mainly diabetes mellitus, because negligible amount always appears in urine, but it is not significant. But yes, if traces, or you can say, you must have seen that biochemical profile plus, that means if sugar is coming in urine, that is strictly a case of diabetes mellitus. And for that, we perform Benedict's test. And if we talk about the composition of Benedict's test, this is also very important. This Benedict's test consists of copper sulfate. It consists of a alkali which is generally weak alkali, and it is sodium carbonate, and it consists of a salt which is sodium potassium tartarate. So this is the composition of Benedict's test, right? So what happens is, in the sugar, you can say in the urine, the predominant monosaccharides are glucose, fructose or galactose. So what will happen is this glucose, fructose or galactose, they have tendency to form enediols. And these enediols are very good reducing agents. They are good reducing agents. On the other hand, what is happening is this copper sulfate on heating is being dissociated into cupric and sulfate ions. Right. So enediols are coming from this and a salt is already there. So what will happen is this cupric ions will be reduced to cuprous, and basically we get red precipitates of cuprous oxide. cuprous oxide. Now the question is that earlier, say some 5, 6 or 10 years back, Fehling's test was also there, which was conducted to distinguish between reducing and non-reducing sugars, but वो test नहीं चला. That test was not, applicable. Why? Because there were certain do's and don'ts in Fehling's test. The most important one was the alkali. In Fehling's test, a very strong alkali is used, which is generally potassium or sodium hydroxide. Now, what is the purpose of this weak alkali? This weak alkali results into proper enolization. The more weaker the alkali, the better enolization will be. Better enolization or better enediol formation, and that is what is our target, because we have to select, we have to basically see how much amount of a carbohydrate is present. So always remember, weak alkali, better enolization. So that is why Fehling's test was discarded. Okay, so now I have mentioned that it is a quantitative test, and that too semi-quantitative test. So what do you mean by the term semi-quantitative? It means that this type of coloration will be there. Just show you the list. See, if we talk about, you can see the variation in the colors. Okay, so what happens is, you have to just remember a mnemonic. Jaise we know rainbow colors ke liye the mnemonic is GYOR. So GYOR. G stands for green, Y stands for yellow, O stands for orange, and R stands for red. Okay, so if we are adding Benedict's reagent in sample of urine, okay, and if we heat it, so if the coloration is red, we can say that the amount of sugar is more than 2%. But if it is orange, we can say it lies between 1.5 to 2.0%. If it is yellow, it is 1.0 to 1.5%, and if it is green, it is 0.5 to 1%. Some amount of sugar automatically will be excreted in urine. Okay, that is why it is known as semi-quantitative test. As you can see here, if it is giving red color, brick red precipitates, the amount of sugar is more. If it is giving orange color, little less, yellow, more less, green is just the person is going towards diabetes, so he can take control of the diet. Okay, so this is a very, very important test which distinguishes between reducing and non-reducing sugars. And the composition is simply copper sulfate is there. You can say a weak alkali is there. The more weaker the alkali is, the better enolization would be. And after that, it consists of a salt. The purpose of adding salt is to prevent the earlier precipitation. So it is like, in a sequential way, cupric ions will be reduced to cuprous, and then they are going to give what? They are going to give a characteristic brick red color, right? So this is Benedict's test. Then Fehling's test, I will just write Fehling's test is not used. Not used. The reason is that strong alkali, so less enediol formation or less enolization is there. And second is, whenever we are, because this is a test which can actually check the presence of abnormal constituents in urine. But Fehling's test mein the only thing is that sometimes uric acid also might interfere, which can give again a wrong result. Might interfere. So this is Benedict's test, right? So now we have come to know the sugar is reducing or non-reducing, and we all know that all sugars are reducing except sucrose and trehalose. This we have already discussed in detail in the chemistry of carbohydrates video. Now after that, now we have to see whether the sugar is an aldehyde or a ketone. So for that, Seliwanoff's test is there. And this Seliwanoff's test is a test given by only ketonic group. Okay, so only by ketohexoses. Example is fructose and sucrose and sucrose. Now, why sucrose? Because again, if we heat sucrose, then it will be converted into fructose and glucose. And we know that this again I have discussed in my video on chemistry of carbohydrates, that sucrose is known as an invert sugar because it appears to be dextrorotatory, but it is levorotatory because of more levorotatory power of fructose. Okay. And if we talk about the test is simple. We can say that we add Seliwanoff's reagent, which is resorcinol in HCl. Resorcinol in concentrated HCl. And we just have to warm the solution. Okay. And there is cherry red colored complex which is produced. So basically, again, if we are talking about HCl, HCl is an oxidizing agent. So it will give initially what it is going to give? It is going to give characteristic furfural, and this furfural will condense in the presence of resorcinol to give a cherry red colored complex. So this will be the color. So if this type of coloration appears, we can say the sample is either fructose or it is a ketohexose basically, or it is having a ketonic group. Okay. So these are the qualitative tests for carbohydrates. We always start with, let's summarize, we always start with Molisch test, which is given by all carbohydrates. When we know that the sample is a carbohydrate, then we perform iodine test. In iodine test, if it is giving a bluish or violetish color, that means it is starch. So we will go for the confirmatory test for starch. But if it is not giving that color, it can be a mono or a disaccharide. To differentiate between them, we carry on Barfoed's test. Barfoed's test may, if precipitation appears immediately, it is a monosaccharide. If delayed precipitation is there, it's a disaccharide. And the most important semi-quantitative test is the Benedict's test, which distinguishes between reducing and non-reducing sugars. Clinically, this test is very, very important because this is one of the most sensitive indicators of diabetes mellitus whenever we have to check the presence of abnormal constituents in urine. Why semi-quantitative? Because again it is going to give a variety of colors, and if it is negative, it will give the color of Benedict's test, which is blue because of copper sulfate. If it is positive, reducing sugar is present. If it is negative, non-reducing sugar is present. And then we come over to the Seliwanoff's test, which is given by only the ketohexoses, because Seliwanoff's test, why it is important? Because glucose and fructose are going to give all the test positive. Suppose if a patient comes to us and he is suffering from fructosuria, okay, no doubt enzyme based studies are also important, but if we have to differentiate, because there is a difference between a patient suffering from fructosuria and a patient suffering from glucosuria. So fructosuria mein Seliwanoff's test will be positive. This is very important because fructosuria ki different etiology, everything is different. It is due to the deficiency of the enzyme fructokinase. Okay. So that is why these qualitative tests also play a very, very important role in checking which carbohydrate is present. So this is all in this video. I hope this test is clear to all of you. Thank you so much.